Okay, in this video, I would like to show my a reviewer that I've made back when I was in 12th grade, um, when I was preparing for the first quarter uh, biology exam. So this was basically about biochemistry, um, glycolysis, pyruvate oxidation, um, what do you call the third part? The third part of the process, citric acid cycle, and then um, we'll recall that eventually. What happens after the citric acid cycle? My memory is a little vague about it as of now. So yeah, one good thing is that my reviewer has the important details. Um, uh, part kind. I also want to make a video for this because this reviewer is very special to me. I placed a lot of effort into making this, although my capabilities aren't limited to just making this single reviewer. Looking back, I wasn't able to prepare for the USTET in the Philippines. That is the University of Santo Tomas entrance test. Because partly I was preparing for this reviewer. I was preparing this reviewer for the exam designated for this. So yeah, let's start officially. So there are two modes of ATP synthesis. What we mean by synthesis is that the creation of ATP. So we have substrate level phosphorylation and oxidative phosphorylation. By the way, um, I'll let you know in advance that all the information in this reviewer, that's the only thing, those are the only things that I will explain. Anything such as, for example, defining the meaning of specific parts, as long as they're not written, I won't be able to explain it anymore. So yeah, you'll have to also consider that if you still want to watch the video. But yeah, let's just have a rundown. I don't remember why I wrote this, but it says if two blank are considered isomers, then they have the same molecular formula, but they have different molecular arrangements. So glycolysis, this is the first step. It's a 10-step process. So how many? Let me just check the back page. Okay, so yeah, there are 4 steps. I mentioned 3 earlier. The last one is the electron transport chain, or chemiosmosis. They're different terms, but I think this is the more appropriate term, chemiosmosis. Glycolysis is the first stage or first process of cellular respiration. There are 2 phases, the energy investment phase and the energy payoff phase. So glucose, 2 ATP use that produces 2 Ps or 2 pyruvates and 4 ADPs. ADP stands for adenosine diphosphate, not tri-di. So next, for the energy payoff phase, 4 ADP and 4 pyruvate creates 4 ATP or adenosine triphosphate, and then 2 NAD plus 4 electrons. I think this means electrons. I'm not sure what this is. 4H plus. This produces 2 pyruvate and 2 water molecules. So the net products of glycolysis is, so for glucose, that produces 2 pyruvate and 2 water molecules. 4 ATP, 4 ATPs are formed here, according here, and 2 ATPs are used. This is from here. So the net, what we have left as a final product is the number of 2 ATPs. 2 NAD plus 4 electrons plus 4 H plus. This produces 2 NADH and 2 H plus. for some reason. I can't really prove why, but best you memorize that. Okay, so okay, let me just adjust the camera. I guess I won't be able to show the entire paper. Let's just start first with this part. So stage 1 is glycolysis again. Glucose is converted into pyruvate through what's called the dehydration process. Sorry, what does that say? Degradation process. 1 glucose into 2 pyruvate. Remember the numbers. 1 glucose, 2 pyruvate. So cytosol, ATP, substrate level phosphorylation. Some steps in glycolysis and citric acid cycle are redox reactions, in which dehydrogenase transfers from substrates to NADH plus 3 times. So this is for steps 3, 4, and 8. The transfer, from substrates to FADH2, this happens 1 time. This is in step 6. What am I referring to when I mentioned which specific steps? These are the steps in the citric acid cycle, not the 10-step glycolysis process. More on that later. So for pyruvate oxidation, 2-acetyl-CoA. So from here, there is a breakdown of glucose to CO2. So CO2 that's produced by cellular respiration represents the fragments of oxidized organic molecules. So as you can see, looking at the figure itself, glycolysis happens outside the mitochondrion. The rest, for steps 2, 3, and 4, they all happen within the mitochondrion. Again, this is inside the cell, responsible for being the powerhouse for generating ATP. And that is the energy for the cell. So going to the citric acid cycle, this is anaerobic, doesn't require, just like chemiosmosis, steps 3 and 4 specifically, they are anaerobic. That means they do not need oxygen to function or to happen. So ATP, substrate level phosphorylation, this is the process in which ATP is formed by the transfer of a phosphate group from substrate to ADP. So I think this is the reason why instead of being adenosine diphosphate, di as in suffix as 2, it becomes ATP, tri, that means as 3. So an addition of a phosphate group. I think that's what's up in this case. So again, more on the citric acid cycle later. I believe I've made a diagram of it later. I'll show it later. So, chemiosmosis. Electron transport chain, this is the 3rd stage of respiration. I'm not sure why it's not called 4th. Anyways, there has to be a reason why. And I recognize that. So, there is an acceptance of electrons from the breakdown of products of stage 1 and 2. So these electrons are passed from one molecule to another. So at the end of the chain, electrons are combined with oxygen and H or hydrogen ions. So this causes the formation of water. I'll be right back. Okay. So the energy released at each chain step is stored in a form that the mitochondrion can use to make ATP from ADP. So the ATP that's from here allows for oxidative phosphorylation. So this is the process in which ATP is formed as a result of redox reactions in the electron transport chain, or ETC. So far, I recognize that I'm not able to explain this properly. Yeah. What does this mean? If only I can recall. Electrons carried via NADH. Electrons carried via NADH and FADH2. Maybe partly the reason why is because only during the citric acid cycle does FADH2 come inside the picture of cellular respiration. As of this part, the glycolysis phase, only NADH is involved. FADH is not mentioned yet. So yeah. Okay, so this is the 10-step process of glycolysis. So again, it is divided into two phases. The energy investment phase, meaning you have to spend ATP. Then you have the energy payoff phase, in which you earn 280. Sorry, let me just make sure. What's the difference between this and this? Because usually the tendency is you have to ask, is the production of ATP actually 2 or 4? Because NADH is the same number, 2 and 2, as you can see here. We'll see. I hope we get the answer later as we go through the steps. But yes, so for the energy investment phase, this consumes energy, like I said. So you'll be spending ATP, what's currently existing in the cell. So this consists of steps 1 to 5. For step 1, we have, so coming from glucose, I'm not sure what the encircled terms are supposed to mean. But yes, from glucose to glucose 6-phosphate, I guess the ingredient in order to carry out the process is hexokinase for step 1. For step 2, this is phosphoglucoisomerase. So from glucose 6-phosphate into fructose 6-phosphate. They are isomers. I'm not sure what being isomers means, but yeah. Feel free to research on that. Step 3, from fructose 6-phosphate to fructose 1,6-biphosphate. What's in the middle is phosphocructokinase. Okay, phosphorylation of this. Oh, okay. Phosphorylation. I think that's the term. So, for steps 1, 3. Okay, there's a reason why I highlighted steps 1, 3, 6, 7, and 10. I think these specific steps involve the process of phosphorylation. So from, for example, fructose 6-phosphate to fructose 1,6-biphosphate, for example, in step 10, from PEP to pyruvate. So yeah. Next step. Wait a minute. So also in the same mentioned steps, I think it's implied that whenever phosphorylation is involved, there is a transfer. In the energy investment phase, it's from ATP to ADP. In the energy payoff phase, it's from ADP to ATP. So it's the other way around. It kind of makes sense because in the energy payoff phase, you're adding, you're trying to produce energy. So from ADP, it has to be ATP as an end product. In the energy investment phase, you use 2 ATP, specifically in steps 1 and 3, in order to generate adenosine diphosphate. Okay, I believe it makes sense. Okay, step 4, fructose 1,6 biphosphate. It says these two guys are isomers. So aldolase. Okay. Step 5, dihydroxyacetone phosphate. It's rarely reversible, but in step 4 it is reversible, as well as in step 2. But yeah, it turns into 2-glyceraldehyde 3-phosphate. Isomerase catalyzes a rearrangement reaction. I think this is the reason why it's rarely reversible. I'm not sure. I can't guarantee that. So in the end of the energy investment phase, there is a production of 2-glyceraldehyde 3-phosphate. So this is, well, I don't think it's necessary to memorize each specific term, but this one, in the last, as the last result of the energy investment phase, I think it's worth memorizing. Okay, so let's go to the energy payoff. So again, this produces energy. This consists of steps 6 to 10. Okay, so for step 6, from 2-NAD+, it becomes 2-NADH. Glyceraldehyde 3-phosphate, it says this step is reversible. It turns into 1,3-biphosphoglycerate. What does this say? Remove what? Not sure what this means. Okay, so sugar is oxidized, turning NAD+ to NADH. Energy is released from the said redox reaction. Energy that's released from the said redox reaction is used to attach the phosphate group to the oxidized substrate, making the product increase potential energy. Okay, it makes sense. So in the oxidation of sugar, a plus is removed, I guess, and in the turning of NAD+ to NADH, you add a plus. I'm not sure. Reducing or turning, I think they're synonymous. This is usually what happens if you make a video about a review you made more than a year later. I should have done this when I was still very familiar with what I was writing. Yeah, missed opportunities. Okay, step 7, substrate level phosphorylation from 2-ADP to 2-ATP. So 1,3-biphosphoglycerate to 3-phosphoglycerate. Also, another question you might ask, generally, what does it mean? What can be possible? And what's the importance for the feature of reversibility in some steps? For example, here you can't reverse. So is there a reason for that? Is there a good reason? But in step 4, as well as in step 2 and 7, you can see there are interchanging arrows. So that means they're reversible. What's the implication? Anyways, going back, for step 8, from 3-phosphoglycerate to 2-phosphoglycerate, what's in the middle is phosphoglyceromutase. I think I can see the reason why we have these encircled terms in the middle. They're catalyzers, if that's even a word. I'm not sure. Step 9. 2-phosphoglycerate to phosphoenol pyruvate or PEP. What's in the middle is enolase. Dehydration reaction happens from enolase to H2O. I'm not sure what that means. By the way, don't forget the units. 2, 2, 2, 2, 2, 2, 2, 2. Step 10. From phosphoenol pyruvate or PEP to pyruvate. What's in the middle is pyruvate kinase. This is also substrate level phosphorylation. I'm not going to consider this anymore. I guess this is the correct one. So the end product is 2 ATP and 2 NADH. Ooh, okay. It makes sense. So in the EI phase, 2 ATPs are spent. So in the energy payoff phase, 4 ATPs are produced. But because we have to account for what was invested earlier, the end product in total is 2 ATP left and 2 NADH. Okay, that makes sense. So what about this? Kinase. So kinase enzyme is involved in the addition of a phosphate group reaction. So I believe this is steps 7 and 10. This is where we get the 2 ATPs, as you can see. 2 and 2. Where do we get the 2 NADH? We get it from step 6. From 2 NAD plus to 2 NADH. In the EI phase, where do we see that the 2 ATPs were spent? Each in step 1 and 3. Okay. What's this? Net reaction. Okay. So glucose plus 2 NAD plus 2 ADP plus what's this? EI. Is this supposed to mean phosphate? I'm not sure. So this equals to the production of 2 pyruvate, 2 NADH, 2 ATP, 2 hydrogens. I'm not sure what this means again. H plus 2 H2O. I'm not sure what's that supposed to imply. Phosphorylation. So this is the addition of a phosphate group to a substrate. Be right back.